Hello, everyone, and welcome to today's webinar, Modeling the Midbrain Floor Plate and Mature Dopaminergic Neurons with Human Pluripotent Stem Cells in 3D. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting as many questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions that we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I would now like to welcome our speaker, Dr. Richard Josephson, Senior Staff Scientist, R&D, Cell Biology at Thermo Fisher Scientific. For a complete biography on our speaker, please visit the presenter tab on the far left of your screen. So without further ado, Dr. Josephson, you may begin your presentation.
Okay, thank you. Hi, I'm Richard Josephson. I'm with Thermo Fisher Scientific and located in Frederick, Maryland in the US. I'm going to talk today about methods to generate mature midbrain organoids and about our efforts to use them for modeling Parkinson's disease in vitro. To give you an outline of the topics in this seminar, first I'm going to talk about the reasons why organoids are valuable for studying human disease and introduce some of the issues that are very common in the field. Then second, we'll talk about how we use 3D culture to improve the brain region-specific differentiation of induced pluripotent stem cells. Third, we also explored different methods of organoid formation in an attempt to make more regular structures with more abundant dopaminergic neurons. And fourth, we demonstrate that the dopaminergic neurons we're generating are reaching functional maturity more quickly in 3D culture than in 2D culture. And finally, I'll touch on our ongoing efforts to measure Parkinson's-like degeneration in organoids. So to get started, what are the advantages of organoids? Mainly in 3D, cells have interaction with more neighbors, including the other cell types of the brain that support neurons. Organoids also allow development of 3D layering and brain-like structures. So those two factors may allow neural populations to better resemble the mature brain and its disease states. However, there's a number of challenges with organoids. Organoid development is typically very slow and a lengthy process with a lot of steps. Some of those steps, such as encapsulation inside extracellular matrix, are very hands-on and therefore have a low throughput. And then finally, organoids are usually less reproducible than 2D cultures. And this is a big challenge if you want to do any kind of quantitative disease modeling. So we've got this process of developing organoids broken up into 4 steps, as you see on this slide. The first is to generate large numbers of undifferentiated pluripotent stem cells in suspension culture. Suspension culture allows you to easily get to large scale, but it also adapts the cells from the pluripotent stage to suspension culture. The second step is to optimize the specification of the floor plate in an attempt to obtain nearly pure midbrain floor plate progenitors. Third is to examine the methods of organoid formation and how they affect expanding the floor plate, and choose the physical method that gives you the best differentiation of dopaminergic neurons. Finally, we want to demonstrate maturation of those neurons and show the relevance as a disease model for Parkinson's. So we start the culture at the pluripotent stem cell stage, and we're using a Gibco medium called stem scale PSC suspension medium to do that. This medium allows stem cells to grow in suspension without microcarriers. Instead, the PSC self-assemble into uniform spheroids. And then the workflow is rather simple, but you can get 5 to 10-fold expansion of the cells in every passage while maintaining their pluripotency over a large number of passages. The rest of the steps are all based on another medium system, the Gibco PSC dopaminergic neuron differentiation kit. This is a set of 3 media. The first media is for specification of floor plate in 2D cultures, followed by a medium for floor plate expansion in a series of passages, first in attached culture and then in suspension culture, as you can see in this graphic to the right. And then the last medium is the maturation media that takes you from the floor plate progenitors out to differentiated dopaminergic neurons in about 5 weeks total. So after this kit was released, we had a number of customer requests to develop a version of this for 3D culture to generate midbrain organoids. And so that's what we set out to do. So these are some experiments that were performed by a colleague of mine here in Frederick, Michael Durr, to initiate the differentiation in 3D. He started with PSC cultures growing in stem scale on a rotating culture platform, then swapped them into the floor plate specification media from the differentiation kit. And what he tested first was whether the specification efficiency was affected by the starting size of the undifferentiated PSC spheroids. At 2 or 3 days after a split in stem scale medium, you get uniform spheres of a diameter that reflecting how many days they've been in culture. And that's very different between day 2 and day 3, as you can see in the table. Day 2 spheroids are about 300 microns in diameter. By day 3, they're approaching 500 microns. Excuse me. So then he switched those into the specification media, day 2 or day 3 spheroids. Whoops. And then at the end he allowed the specification to go for 6 or 8 or 10 days in rotating culture. And then for each of those 6 or 8 or 10 day cultures, he swapped into expansion medium up until day 11 in suspension culture, as you see in the graphic here. So then we have spheroids like you see on the right that look very different from the starting PSC spheroids on the left. Each one is ringed by a translucent layer of epithelium that has grown out during the expansion stage. At day 11, these spheroids were passaged and replated onto poly-D lysine and laminin. So those cells dissociated at day 11 were then stained for the midbrain floor plate markers OTX2 and FOXA2. What we found is that the smaller day 2 PSC spheroids were specified with very high efficiency. We got more than 90% positive cells for OTX2 and FOXA2 after just 6 days of specification. Meanwhile, the larger spheres at day 3 after passage showed a significant drop in the floor plate specification efficiency, telling you that initial spheroid diameter is extremely important in responding to these induction cues. For the smaller 2-day spheroids, that 6-day specification turned out to be just as efficient as a 10-day specification as laid out in the original 2D protocol. We also compared the results of that 6-day specification directly with specification in 2D. And what we found is that the efficiency of floor plate induction in 3D, over 90% in 6 days, isn't fully equivalent to specification in 2D. as you can see by the greater than 90% OTX2 FOXA2 positive score. However, there is a difference in the maturation of those dopaminergic neurons from these progenitors. On the right here are images taken from two distinct iPSC lines. In both cases, the neurons positive for tyrosine hydroxylase, TH, and FOXA2 emerged earlier after 3D specification than they had in the 2D protocol. So as you can see on the right, the cultures at day 25 on the far right look very similar to the cultures at day 35 of the 2D protocol. So this difference is due to the specification in 3D because the maturation phase was an attached culture for both. So in a little bit, I'm going to show you how this 3D culture affected the neuronal function in these experiments. First, I want to summarize these initial experiments, showing that we can use the 2D dopaminergic neuron differentiation kit to generate floor plate in 3D. So our starting material is PSC spheroids grown in suspension. The uniform small size turns out to be crucial for efficient specification. And that specification happens more quickly in 3D, 6 days, versus 2D at 10 days. What we achieve in that 6 days is greater than 90% midbrain floor plate. Furthermore, the specification in 3D allows the neurons to mature sooner, taking about 10 days off of the 2D protocol so that you get something similar at 25 days versus 35 days. So for the remainder of this talk, we're going to be following this accelerated timing for 3D differentiation, which is specification for 6 days, expansion for 5 days, and then continue the dopaminergic neuron maturation in rotating vessels for up to 70 days. As you can see in this graphic, it compresses the timeline from the 2D protocol. Next, we're going to examine the formation of organoids in different physical environments, whether the floor plate specification is best done in a shaking culture or static culture, and how adding extracellular matrix will affect the outcome. So first, we tested two different methods of initiating organoids. One is to seed PSC into rotating plates, where embryoid bodies or collections of PSCs self-assemble, and you get many small spheroids per well of, say, a 6-well plate. The other is to do a static culture, where singularized PSC are forced to aggregate into a single embryoid body per well of a Nunc Sphera 96 well U-bottom plate, as cartoonishly drawn here. The Nunc Sphera plates are a good way to produce uniform spheroids, as shown in this illustration at the bottom. Excuse me. It typically takes, well, this cartoon shows it taking 2 days to form an EB, but in reality, we get a single spheroid within 12 hours of adding singularized cells to the Nunc Sphera plate. And then we can swap the PSC suspension media for the floor plate specification media the next day and begin the differentiation. As you see on the lower right, the size of these spheroids is proportional to the number of cells seeded per each well. And furthermore, the diameter of the spheroids varies very little from well to well. Finally, both of these conditions on this slide, the self-assembled EBs and the aggregated EBs, are labeled as naked because I'm not adding extracellular matrix into this specification. So this is what the spheroids looked like during the process of specification, expansion, maturation in these two different physical parameters. So again, this is the protocol with 6 days of specification followed by 5 days expansion. In the rotating culture, the boxes on the left, we start from very small spheroids 2 days post-passage. The specification media for 5 days leads to strong epithelial outgrowth, followed by a general size increase. And what we get by about 3 weeks is very irregular organoids. You can see these sort of pear-shaped structures in the photo labeled day 26, with a lot of, again, that translucent edge around them showing epithelia. In contrast, when we seed dissociated cells into the microwell plate, they aggregate in less than 24 hours. Then we start specification. We get larger and more uniform spheroids 5 days later at the end of specification in the static plate, which tend to become irregular during expansion as there's a great epithelial outgrowth around the perimeter. And then by 3 weeks, we get these kind of irregular looking organoids. They're not too different in size or shape from what we got in the rotating culture. We also introduced extracellular matrix into these two protocols. So a common method of organoid formation is to encapsulate the early organoid in matrigel or gel tracks, both of which are secreted extracellular matrix from a mouse tumor cell line. And what we did is, for the self-assembled spheroids, we encapsulated in Geltrex extracellular matrix at day 2 during the process of floor plate specification. Or because that encapsulation protocol has very low throughput, with the 96 well plates, we added a dilute solution of 2% Geltrex to the micro well plates at day 2. That's 2% in the floor plate specification medium. And one thing I'll note is we cannot cross over with the two methods. We can't encapsulate completely the spheroids in the 96 well plate because then you can't recover them from the wells. And we can't use the diluted Geltrex in the shaking plate because there's a spot in the center of each well with very low rotation speed, and the spheroids will tend to aggregate there due to the stickiness of the extracellular matrix. So let's look at the effect of adding extracellular matrix. On the lower left, we see the results of adding encapsulation to the free-floating spheroids. It leads to a big increase in epithelial outgrowth by day 5. And you can see this in day 10. The organoids are surrounded by a ruffle of outgrowing epithelium. This produces very large and very irregular organoids by 3 weeks, as you see in the image labeled day 26. In contrast, when we add dilute 2% Geltrex to microwells, it produces a similar but more modest epithelial outgrowth, which you can see in this image labeled day 10. There is a ruffle of epithelium, but it doesn't extend as far as it tends to from the encapsulation. However, the size and shape of these organoids formed in the microwells with 2% Geltrex is much more uniform than in the other conditions. Now, what we want to know, of course, is what do the dopaminergic neurons look like in these different methods? But before I can show you that, I have to make a slight digression to talk about tissue clearing. We need to use immunofluorescence, especially for tyrosine hydroxylase, to detect dopaminergic neurons in the organoids. But the thickness of an organoid scatters light, which makes it very difficult to detect cells that are not on the surface. So we use the Invitrogen Cytovista clearing reagent, which reduces light scatter, as you can see in these images on the left. making it possible to image cells inside a cell spheroid. In these images on this slide are all spheroids of tumor cells. I believe they're A549. But this shows how you can visualize and count more cells in a cleared spheroid than an uncleared spheroid. Here, the cleared spheroid is labeled 151 cells detected, whereas uncleared, the same spheroid, you could only detect 57 cells with what's a vital dye here. So it allows you to visualize more cells. As you can see on the right, the cleared spheroids allow you to visualize into the center of a spheroid. So what I did was to fix and permeabilize my midbrain organoids at 3 weeks of differentiation or later, then added the antibodies to detect their targets, and then finally cleared after the antibody incubation. And I'll show you a comparison of cleared and uncleared organoids on the next slide. So here's some examples of midbrain organoids showing you how clearing improves immunofluorescent detection. All three of these images are maximum intensity projections that are compiled from many optical planes imaged on the CX7 laser confocal instrument. The leftmost image is an uncleared 3-week-old organoid that's been stained for tyrosine hydroxylase and MAP2. The center is another 3-week-old organoid stained with the same antibodies and then subsequently cleared with the cytovista reagent. And at right is a 3-week organoid that's been stained for the ventral midbrain markers FOXA2 and OTX2 and then cleared, showing you the large numbers of midbrain floor plate progenitors in these organoids. So now to use this technique to compare the different physical environments for generating the organoids. Again, these images are maximum intensity projections of 3-week-old organoids stained for MAP2 and TH. The MAP2 is in red, TH is in green. So first on the left here, the organoid currently on the left was formed in rotating culture without added extracellular matrix. And it has a fairly simple structure. You can see the outside is ringed with MAP2 positive neurons. It's more clear if you look below the maximum intensity projection at the top to a single optical plane at the bottom. And you only see the red MAP2 staining around the periphery of the organoid. Meanwhile, organoids that were formed in the static U-bottom well, again without extracellular matrix, look like the one on the right. It has more TH positive neurons and more extensive neurites. Now let's look at the effect of adding extracellular matrix during specification. Oops. So, there we go. So when the stirred organoids are encapsulated in Geltrex, it produces a very convoluted epithelial layer, which you can especially see in the single imaging plane at the bottom and picked out with the DAPI stain for nuclei. You get these epithelia that fold back on each other quite a bit, like in the brain. NNAs also have more abundant MAP2 positive neurons marked in red and large patches of TH positive dopaminergic neurons. Meanwhile, if we add dilute Geltrex to the microwell organoids, we get a simpler looking organoid with a thick convoluted layer of TH positive neurons around the outside. And if you look at the bottom right, we have the single plane image showing you that the TH positive neurons are occurring not only at the surface, but also more deeply within the organoid. But these organoids are also rounder and more compact than the organoids formed by the other methods. So we found that extracellular matrix encapsulation increased the complexity of our organoids and the yield of dopaminergic neurons. But using a dilute Geltrex solution in the microwell gave a similar result, but with organoids of regular size, and also allowing a higher throughput than encapsulation does. So to summarize this portion, we've tested 4 environments for organoid formation: rotating or static, no extracellular matrix, or with Geltrex molecules added. All of these methods gave organoids that had dopaminergic neurons inside of 3 weeks of development. However, microwells made the epithelial morphology more complex and improved the yield of dopaminergic neurons. And on top of that, extracellular matrix triggered epithelial outgrowth. Encapsulation led to highly folded epithelia and abundant dopaminergic neurons in some areas of the organoids, whereas the combined microwell and dilute extracellular matrix gave a folded epithelial structure with more evenly distributed dopaminergic neurons. So that last method, the microwell plus ECM, appears to be the most promising. This was confirmed by something that surprised me. Let me take a moment to tell you about neuromelanin. So neuromelanin is a pigment which is found in human dopaminergic neurons of the substantia nigra pars compacta. Loss of that neuromelanin pigment correlates with dopaminergic neuron loss in Parkinson's. As you can see at the image here, these are midbrain sections taken post-mortem. The P labels a section from a Parkinson's patient, and the C is a normal control. And you can see that the Parkinson's patient has lost the neuromelanin staining from the substantia nigra pars compacta. Now, neuromelanin is synthesized from dopamine or from L-DOPA by more than one pathway. So it's an indicator of neurotransmitter synthesis. Neuromelanin pigment has also been reported to occur in human midbrain organoids. In a number of papers, it typically appears after 70 to 100 days of culture, so quite mature organoids. We also observed this reddish-brown neuromelanin pigment in encapsulated organoids, which is the set of pictures at the center of this slide. Typically, after 9 or 10 weeks of culture, we see these clusters of reddish-brown spots. It can be seen in the unencapsulated organoids formed in microwells or free-floating as well, but it's rare, and usually there are only a few spots per organoid, as you can see in the day 63 images on the left. However, we found that surprisingly, the dilute 2% Geltrex addition had a similar effect on neuromelanin as encapsulation. We got neuromelanin in the microwell organoids. But one difference is that we started to observe the pigment in as little as 4 weeks using the microwell method with dilute ECM. And it also occurs in almost every organoid, as you can see in the pictures at the bottom right. Another feature of neuromelanin is that we can use its expression to optimize ingredient concentrations for floor plate specification. So this is a series of organoids that were formed by the microwell 2% Geltrex method. But during specification, we varied the concentration of one of the inducing molecules in the specification mix from 1x up to 6x. And what we saw is that the amount of neuromelanin at 5 weeks increases with increasing inducer concentration up to 4x the typical concentration. And this is easily seen without any need to fix or stain the organoids. We just look for the pigment. In this slide, I've paired each of these with a 3-week organoid generated by the same concentration of inducer, but stained for tyrosine hydroxylase and MAP2 at 3D at 3 weeks. And what we find is the increase in neuromelanin up to 4x correlates with an increase in tyrosine hydroxylase positive neurons at 3 weeks. In addition, we found that this optimization is not changing the regional specification of the organoids. So at the bottom here, we have higher resolution images showing that the Th positive neurons marked in red are co-localized with the GERK2 staining in green. GERK2 is a calcium channel that is a marker of the A9 population of dopaminergic neurons, the same population which is lost in Parkinson's disease. Furthermore, we can also use neuromelanin to optimize the maintenance medium for our organoids. So in these experiments, we started with microwell organoids given dilute Geltrex, as seen at day 0, using the maturation from the maturation medium from the DA kit, which is the leftmost image on the bottom, or adding to that DA maturation supplements that promote neuronal health, B27 in the middle picture or B27 plus in the right picture. And we found that adding either of these health promoting supplements gives a strong increase in neuromelanin by 5 weeks. We suspect that it's stabilizing the neuronal population by its presence of antioxidants, as well as other vitamins and such in these supplements. So to summarize, the extracellular matrix addition, during specification promotes earlier neuromelanin deposition. The proneuronal supplements given during maturation promote stronger neuromelanin synthesis, as you see here. And the abundance of neuromelanin suggests that we're getting earlier maturation of dopaminergic neurons in these conditions. So next, we want to look at the functional maturation of our dopaminergic neurons. To do this, we use a multi-electrode array, or MEA, to measure field potentials surrounding plated cells. An MEA can detect individual action potentials as well as coordinated network spikes across spatially separated electrodes. Typically, for neural organoid work, the organoid is dissociated early on to allow neurons to detach to the MEA individually. You can attach whole organoids to an MEA, but it typically takes, it's difficult to do, and it typically takes at least 1 to 2 weeks before you can read the field potentials, during which you are very carefully fluid changing so as not to disturb that organoid sitting on the electrodes. So let me show you some data, first from dissociated neurons. So this is an image of the DA neurons that were singularized at the end of the expansion stage by my colleague Michael, day 11 of the protocol. And he used the multi-electrode array to compare neurons that have been specified in 2D versus those specified in 3D. So these two plots on the right-hand side, top row, these are called raster plots. Each action potential that the MEA detects is a vertical black bar. Where there are action potentials that are simultaneous across multiple electrodes, those are called out by changing the vertical bars color to blue and adding these purple outline boxes that show you coordination of activity across space. So in these two raster plots, the one at the middle of the slide is the 2D protocol. We are reading at the activity at day 42 of differentiation. So that's the longer specification and expansion, followed by 21 days of maturation. The upper right image is cells that were specified in 3D. So the protocol is shorter. This is day 32 of overall differentiation, but again, 21 days of maturation. And we can see that these two populations are both active by 21 days of maturation. We see coordination across multiple electrodes, telling you that networks are being formed. But if you look at the graphs at the bottom, neurons that were specified in 3D are marked with blue lines. Those specified in 2D are marked with gray lines. Pardon me a moment. What we see is that 3D specification yields higher mean firing rates in the leftmost graph, greater synchrony between electrodes in the middle graph, and a larger number of active electrodes, meaning coordination over larger space in the right graph. So this tells you that specification in 3D has given a boost to neuronal maturation. We're not only getting more active neurons, but they're becoming active earlier in the overall process. Excuse me. So next I want to show you that an MEA array can also measure dopaminergic neuron activity in an intact organoid. So these are images of midbrain organoids that were plated at day 21 of differentiation onto the multi-electrode array, one per well. Within a week after plating, we can see some spontaneous action potentials. I haven't shown you that data. But by 2 weeks after plating, or day 35, we start to see not only individual action potentials, but some coordination between neurons. 2 weeks after that, at day 51, action potentials are much more frequent, and we get much greater coordination, simultaneous firing of action potentials across multiple electrodes marked by these vertical pink lines. The coordination between electrodes is silenced by adding excess dopamine to the medium. That's because adding dopamine suppresses dopaminergic neuronal activity via the D2 autoreceptor. When you wash out the dopamine in the graphs on the right, it restores the activity and the coordination between distant neurons. So this demonstrates that midbrain organoids contain a network of interacting active dopaminergic neurons. So to summarize this section, I want to draw your attention to a few points, mainly on the bottom of the slide. We found that organoid architecture is more complex after formation in microwells or with encapsulation in extracellular matrix. Both of those conditions appeared to increase the numbers of dopaminergic neurons per organoid, but the combination of seeding in microwells with a dilute extracellular matrix produced regularly sized and shaped organoids with abundant Th-positive neurons. Those microwell organoids with ECM also showed evidence of accelerated dopaminergic maturation. We had earlier neuromelanin deposition, as I've shown you, and more consistent electrical activity than the free-floating organoids. I have not shown you that data. But in the last few slides, I want to switch to talking about our work on disease modeling in 2D or 3D. So there are a number of genetic mutations that are associated with familial or sporadic Parkinson's disease. Several of these mutations occur in the genes for alpha-synuclein, SNCA, or LRRK2, which is leucine-rich repeat kinase 2 protein. To study the effects of these mutations in isolation, the Thermo Fisher Customer Services team introduced these mutations into iPSC lines. using CRISPR-Cas9 and homology-directed repair. Then the edited cells were sorted live to obtain clonal lines that are heterozygous or homozygous for the mutations or unchanged wild type. And as you can see in the pie charts at the bottom, after introducing a mutation into LRRK2 or alpha-synuclein, anywhere from 4% to 48% of clones carried the homozygous mutation. We then wanted to test whether the homozygous mutations in these genes increase apoptosis in dopaminergic neurons. To do so, we're going to measure apoptosis using the cell event caspase 3,7 detection reagent. This is a probe that is activated by cleavage by caspase 3 or caspase 7, which allows it to bind to DNA and fluoresce. So in the images on the right of this slide, we see spheroids made up of A549 cells that have been treated with different concentrations of the drug niclosamide, which blocks oxidative phosphorylation. We also see that the green fluorescence from the cell event probe increases with the drug concentration. And we can measure that fluorescence with a plate reader or a high content imager to produce a sigmoidal toxicity curve, as you see in the lower right. So then the custom services team used the cell event caspase 3,7 probe to test the effects of those introduced mutations on 2D cultures of PSC-derived dopaminergic neurons. So in these images on the left, staurosporin or 6-hydroxydopamine both increased apoptosis in neurons. And this was detected by the green fluorescence of the cell event probe. On the right, the graphs show that the LRRK2 mutant neurons, which is the second population from the left, show increased sensitivity to 6-hydroxydopamine. Whereas the alpha-synuclein mutant cells, the heterozygotes, are the rightmost population in these graphs, are sensitive to 6-hydroxydopamine only when they're starved for glucose. Now, we're also using the same mutant and control lines to look at cell death in 3D generated organoids. But our first 3D test was to grow out the PSC spheroids. And then we exposed these to various doses of the toxin staurosporin, which triggers apoptosis, in the presence of the cell event probe for 5 hours. At the bottom, you can see that the cell event fluorescence increases with the dose of staurosporin, and we can measure it very easily just using a Varioscan plate reader to give us sigmoidal response curves. This suggests that the cell event assay is sensitive enough to use in 3D. So next, we hope to use it to measure cell death in midbrain organoids, and hopefully cell death that is specific to dopaminergic neurons, as it is in Parkinson's. So to do this, we're going to use 6-hydroxydopamine, which is a dopamine analog that is specifically taken up by dopaminergic neurons and specifically damages those neurons. And we're doing these experiments on mature 6-week-old midbrain organoids. We treat with the 6-hydroxydopamine for 48 hours, then come back 2 days later and add the cell event caspase probe. Then after an hour, fix the probe products in place and conduct immunofluorescence to detect TH to find our dopaminergic neurons. What we'd like to see is both an increase in the cell event fluorescence with an increase in the 6-hydroxydopamine dose and a decrease in the number of surviving TH-positive dopaminergic neurons. However, it turns out to be very difficult to make these measurements in 6-week organoids. There's a couple of example images of organoids at the right here. One that was treated with 25 micromolar 6-hydroxydopamine and another treated with 300 micromolar. These organoids are very large, greater than a millimeter in diameter, and dense, so that the penetration of light is not complete even after clearing. The clearing is improved by serial dehydration, but that tends to strongly distort these large organoids. Makes them look a little like a raisin. What we found was that the fluorescence of the cell event dye did not increase with the dosage of 6-hydroxydopamine. And I believe the reason for this is that there's a very high background signal, mainly on the outer surface of these organoids. That's because, for one thing, in all neuronal cultures, you have cell death that's continually occurring hand in hand with the maturation, particularly among neurons that fail to form synaptic connections. And secondly, organoids are frequently reported to be showing increasing stress during lengthening culture. So both of those factors increase the number of apoptotic cells, even in the untreated organoids. So we're left with either relying on the TH antibody to try to count surviving dopaminergic neurons. And you can see there are more here at 25 micromolar than there are at 300 micromolar. Or try to co-localize the green dye of the caspase probe with the TH staining. But the other issue that we have is the numbers of TH positive neurons still vary quite a bit from organoid to organoid. So we don't have an even baseline. That means we need to image large numbers of organoids in order to try to get reproducible data. And we really need more automated imaging and more automated analysis to get that kind of large numbers of data that will even out our toxicity curves. However, it's possible to make some adjustments to the assay to improve these results. So this slide shows two experiments that were done with the wild type and alpha-synuclein mutant organoids. In both experiments, they are exposed to rotenone, which increases oxidative stress by inhibiting mitochondrial complex 1. At the left, we have 3-week organoids that are stained with the cell event probe and the TH antibody. And at 3 weeks, we can see extensive damage is done by the rotenone to the TH positive neurites, the purple stained cells in the mutant line specifically. But the green dye shows that the background for cell event is still high, but here the number of puncta, of green puncta, does increase with the dose of rotenone for both the mutant and the wild type. But we were unable to collect enough data to really put these into a sensitivity curve. At the right is an experiment done with 5-week organoids that were exposed to rotenone and then cryosectioned. These were then stained with antibodies to TH and an antibody to activated caspase 3. And in these cryosections, we could see that when given the higher rotenone dose, the 25 micromolar at the far right of the slide, the SINCA mutant TH positive neurons that appear in green at the lower right are associated with activated caspase 3, which shows up as the red staining in that image. Whereas the wild type organoid shows much less activated caspase associated with those green TH positive neurons. So obviously we would prefer to use the more mature organoids for disease modeling because Parkinson's mainly affects older brains. And we want at least to have long established neurons with networked electrical activity in our disease model. However, the younger neurons give us a more linear response to the toxins. There's several ways that we could improve these assays. One is we want to increase the signal by having larger numbers of dopaminergic neurons per organoid, while also decreasing the noise by one, identifying the correct assay to run, the correct readout, whether that's antibody or Cas-based probe, and by reducing the stress that these organoids are under in culture. Another factor is that to do any kind of drug discovery with this disease model, we need high throughput. And high throughput is simply not obtainable by doing cryosectioning and antibody staining. There are high throughput compatible viability assays, but those are not, that viability dye is not going to be specific to the dopaminergic neurons. So we would need to reduce the noise level of spontaneous death of every other cell type in the organoid. So if we want to do screening, it may be more suitable to run a functional assay. So for example, as I showed you, the neuromelanin production is easy to observe in live organoids. So that could be an output. Dopamine release is relatively easy to measure if you have the right HPLC equipment. And the electrical activity, the electrical output and coordination of the dopaminergic neurons can be measured by an MEA. While it's difficult to place these organoids onto a flat MEA, there are reports of new MEA technologies that can grip organoids or penetrate the organoids to record a larger fraction of the neurons within each one. But ultimately, it's the users out there, all of you, who are going to be choosing the assays that are most appropriate for your experiments. Our focus right now is to improve the health and consistency of midbrain organoids and try to push past all the barriers to making mature brain models. So to summarize, where this work is at, we've shown that the reagents and media that were designed to direct pluripotent stem cells to midbrain fates in 2D can be used to generate midbrain organoids in 3D. And in fact, floor plate specification occurs more quickly in 3D. The signaling molecules that we've used can be further optimized to increase the proportion of dopaminergic neurons in organoids. And also, culturing in 3D accelerates the maturation of midbrain dopaminergic neurons, giving us earlier electrical activity and stronger networking. Furthermore, the maturation is influenced by the physical environment during the floor plate specification. As I showed you, the microwells with dilute matrix accelerates neurotransmitter synthesis and gives earlier electrical activity. Finally, modeling Parkinson's disease is possible in these more brain-like 3D structures. We can see the effect of a disease-linked mutation in alpha-synuclein. But we do have to use very low throughput assays right now and rely on antibody staining and even cryosectioning. Because higher throughput assays are currently less specific to dopaminergic neurons. Our goals are to increase the signal by optimizing the numbers and distribution of healthy dopaminergic neurons in each organoid and decrease the noise by reducing cell stress. So with that, I would like to thank all my colleagues in Thermo Fisher Scientific who've supplied cell lines or data for this presentation, and also those in Thermo and at LabRoots who organized this seminar, and all of you for your attention. Thank you very much, and I would be happy to take any questions right now. Thank you.
Dr. Josephson, thank you for that excellent presentation. Such detailed explanations you gave. It's wonderful. Well, I'm excited to say that it is now time for our live Q&A portion of our webinar. So to our audience, if you haven't submitted any questions yet, please do so now. Just type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So Dr. Josephson, we've got quite a few questions that have already come in. So I'm going to just dive right in. Let's start with this first one. What's the difference between a spheroid and an organoid?
There are multiple definitions. how to distinguish the two. For some people use the simple definition of an organoid that it's any 3D structure derived from pluripotent stem cells. I prefer to define it more in terms of the complexity. So the spheroids that I showed you were typically made up of a single cell type, like a A549 tumor cell, and they're very homogeneous. All the cells are essentially alike. Whereas an organoid is self-organizing. They have layers. It has different cell types. So one thing that's really simple is distinguishing between a tumor cell spheroid is like the ones I showed you here. None of the cells are distinguished from each other, and a tumoroid, which actually has an epithelial barrier and different cell types within it. So complexity is the difference.
Excellent. Thank you for that description. Next question. How do you refresh the medium containing free-floating spheroids without losing them?
Very carefully. The way that I have been doing it, particularly for the 96 well plates, is first of all, you don't change all of the medium at once. Every medium change is half of the medium being replaced at a time, or 2/3 of the medium being replaced at a time. You can do that slowly and carefully with a multi-channel pipette. But there's also colleagues of mine have found it can be done in a more automated fashion using a plate washer. Let me bring up this one slide that shows you what this looks like. So in the lower right is an image of the Thermo Scientific well wash microplate washer. It's meant to wash like antibody assays out of multiple wells, but they were able to use it to change half the media at a time in multiple 96 well plates of spheroids with a very low rate of loss. So that's one way, if you're scaling up, that you can do this more easily.
Excellent. Now, we've had some questions come in regarding throughput. How can you improve or increase throughput for imaging?
For imaging, well, it's all dependent on your equipment, basically. So most of the images I'm doing here were taken with the CX7 laser confocal, which, while it's a very detailed, high-resolution system, it takes a long time to collect images. If you can afford one, a light sheet microscope will generally be a much faster way of collecting images. But there's also an improvement that's been made to the CX7 laser system. That's the CX7 Laser Pro, which, let me show you a slide I was given about that. This is a newer generation of this high content screening platform. And one thing that it has is this Eureka Scan Finder software that it makes it faster for you to find these spheroids and image them by first doing a pass at low resolution or low magnification, finding the organoids for you, and then going back and imaging at higher magnification.
Now let's stay on the topic. Oh, continue.
Sorry, no, go ahead.
Well, I just wanted to stay on the topic of throughput. Now, is there a higher throughput alternative to Sphera 96 well plates?
Well, we haven't done too much experimenting with it. As I said, we haven't been able to use the dilute gel tricks in a horizontally shaking platform because it brings the spheroids together and aggregates them. But we are looking into whether a bioreactor, especially a horizontal wheel bioreactor, might be a good way to scale up because it doesn't have that dead spot that a rotating plate does. So it's keeping the spheroids in constant motion, and that should keep them from aggregating, but don't take my word for it.
Let's jump to our next question. How important is organoid shape, regularity, and sphericity in downstream DM maturation?
I don't think it's very important for the maturation. We get good maturation from really odd-looking organoids, but the regular size and shape is very helpful for downstream assays. So you don't want to have variation between, say, some 500 micron organoids comparing them to 1 millimeter organoids. It's just not going to be very comparable.
We have time for one more question. Is there a recommended optimal size of neuroorganoid for passaging and imaging?
So for these organoids, I never passage them. From the moment they're formed as a spheroid as PSC all the way out to 10 weeks later, they're not passaged because unfortunately you can dissociate them up to about 3 weeks and replate them onto an MEA, but beyond that you'll be killing the neurons. The optimal size for imaging, though, is generally less than a millimeter. Because of diffusion limits, anything bigger than maybe 800 microns is going to have dead cells at the core. So that's kind of useless to you. And the penetration of imaging that you can get with the site of Vista is quite a bit more than that. So millimeter is not too big, but you want something you can look at under high magnification in as few fields as possible. So again, as a matter of throughput, you want to try to keep them below a millimeter.
Noted. Well, thank you so much, Dr. Josephson. What a delightful webinar this has been. Do you have any final comments for our audience?
Well, all I can say is if you want to try these protocols for yourself, there is a Thermo Fisher website with some help. It's thermofisher.com/3D models. We have a 3D cell culture handbook there and some digital courses and probes handbook and number of protocols. I am not sure whether the protocol that I used for this is on that website. It ought to be, but I could not find it today. If you cannot find any of these protocols, reach out to your Thermo Fisher sales reps and they will get them to you.
Excellent. And Dr. Josephson, if you just jump to slide 41, push that to the audience. That link is on there so we can just get it in front of our audience. So in case anybody wants to go there or snap a picture, we'll get that in front of their face right now. So excellent. Well, I want to thank you again, Dr. Josephson, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Now, before you go, I do want to remind everybody that today's webcast can be viewed on demand, and LabRoots will send you an email when it's available for replay. So be sure to share that with your friends and colleagues who may have missed today's live event. I also want to say we've had some great questions that came in, but due to time, we were unable to answer the rest of them. So your questions that we did not answer and those that are submitted during the on-demand period, they will be answered via that email address you provided at the time of registration. So everyone, thanks for joining us. Hope to see you again soon. Have a great day.
Thank you.
Thank you.